Right now, I will be explaining what is SDS page. SDS page, which stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis, which is an analytical technique used to separate proteins based on their molecular weight. Meanwhile, electrophoresis is a technique used to separate macromolecules in an electrical field. This technique employs discontinuous polyacrylamide gel for support and SDS for denaturation. This technique developed by Ulrich K. Lemley in 1970. Right now, I will be explaining what is the application of SDS page. SDS page mainly used for research tool. It can measure molecular weight, peptide mapping, protein identification, determination of sample purity, and identifying disulfide bond. This is principle of SDS page. SDS, sodium dodecyl sulfate, is an anionic detergent. When SDS is added to a solution containing protein, the detergent molecules bind with the polypeptide of the protein. Thus, number of SDS molecule bound to the protein is nearly half of the number of amino acid residues in the protein. The SDS protein complex have some net negative charges. Therefore, protein move towards. They are not depending on their size. Mobility of the protein molecule depends on the net charge, size and shape of the molecule. Type of samples that can be used in SDS page include cell lysates, purified proteins, recombinant proteins, tissue extracts, and serum and plasma.
Now I will explain about the sample preparation. Before running the SDS page, the protein sample must be prepared to ensure accurate separation. In this demonstration, a small piece of fish muscle is used as the protein source. First, the tissue is placed into a microcentrifuge tube containing Lemley's sample buffer. This buffer contains SDS, which unfolds the proteins and coats them with a uniform negative charge. It is also contains a reducing agent that breaks disulfide bonds, allowing the proteins to become completely denatured. Next, the tube is gently mixed to ensure the tissue is fully exposed to the buffer. The sample is then left at room temperature for a few minutes so that the buffer can penetrate the tissue and begin extracting the proteins. After incubation, the liquid containing the extracted protein is carefully transferred into a clean tube while avoiding any solid tissue fragments. The sample is then heated at 95 degrees Celsius for 5 minutes. This heating step completes protein denaturation, ensuring that proteins are separated according to their molecular weight during electrophoresis. At this stage, the protein sample is fully prepared and can either be used immediately for SDS page or stored at -20 degrees Celsius for further analysis. Next, I will explain the gel casting process for SDS page. The first step is to prepare the glass plates. Make sure both plates are clean and completely dry to prevent contamination or leakage. Then, place the spacer plate together with the short glass plate into the casting frame. Check that both plates are aligned evenly before locking them securely with the pressure caps. Next, insert the comb into the cassette and mark the glass plate about 5 mm below the comb. These marks serve as the filling level for the resolving gel. After that, place the assembled cassette into the casting stand and ensure it tightly sealed. Now, prepare the resolving gel solution by mixing the required acrylamide solution with 10 ml and freshly prepared 10% ABS. Mix the solution gently to avoid introducing air bubbles, then slowly pour it into the cassette until it reaches the marked level. Without waiting for the resolving gel to fully polymerize, prepare the stacking gel solution using the same approach. Once it is ready, pour it carefully on top of the resolving gel and immediately insert the comb, making sure that no air bubbles are trapped beneath the comb tip. Finally, allow the gel to polymerize for at least 30 minutes. After solidification, the gel is ready for protein sample loading and SDS page electrophoresis.
Hi everyone, my name is Aisha. Now let's proceed with the electrophoresis. Before electrophoresis, the polyacrylamide gel is prepared through the polymerization of acrylamide and cross-linking by bis-acrylamide. This process forms a gel structure with many tiny pores that act as a molecule filter. So this size of pores can be adjusted by changing the concentration of polyacrylamide. After the gel is prepared, the cassette is located between two electrodes. The positive electrode is connected to the lower part of the gel and negative electrode to the upper part of the gel. Next, the gel is placed inside a chamber filled with running buffer to ensure the conduction of the current. Next, the protein sample and the molecular weight marker is loaded into separate well using micropipette. This protein sample contains bromophenol blue and also glycerol. And lastly, the lead is then connected to the power supply. Alright, now, when the electric current is applied, the negatively charged protein will move towards the positive electrode. Since all the proteins are treated with SDS, they all have uniform negative charge. So they are separated primarily according to their molecular weight. Therefore, smaller protein will move quickly through the gel and travel farther compared to the larger proteins. Bear in mind that there is two parts of the gel, which is the stacking gel and separating gel. The stacking gel is the upper part of the gel and has larger pores that function to gather all the proteins into a thin band before the separation process. During this process, the chloride ions move faster and act as the leading ions, meanwhile the glycine ion moves slower and act as the trailing ion. As a result, the protein samples are trapped between these two ions and line up evenly before entering the separating gel. And now, the electrophoresis process continues until the tracking die reaches the bottom of the gel. At this point, the power supply is turned off and the gel is removed for staining and visualization steps.
After protein have been separated according to their molecular weight using SDS-Page, the next step is Western blotting. Although SDS-Page separates protein based on their size, it cannot identify a specific protein. Therefore, Western blotting is used to specifically detect the protein of interest using antibody. The first step is protein transfer, where the proteins are transferred from the polyacrylamide gel onto a polyvinylidene fluoride PVDF membrane using an electric current. This process, known as electroblotting, preserves the protein pattern while providing a stable surface for antibody detection. Next, the membrane is blocked using a blocking solution such as non-felmite or bovine serum albumin, also known as BSA. This blocks the empty spaces on the membrane and prevents the non-specific antibody binding, reducing the background signal. The membrane is then incubated with the primary antibody, which binds specifically to the target protein. After that, the membrane is washed to remove any unbound primary antibodies, ensuring that only specific binding remains. Next, a secondary antibody is added. It binds to the primary antibody and carries an enzyme called horseradish peroxidase or HRP. The membrane is washed again to remove any excess secondary antibodies. Finally, a detection region is added. The HRP enzyme attached to the secondary antibody reacts with the substrate to produce a visible signal, allowing the target protein to be detected on the membrane. The appearance of a protein band confirms that the target protein has been successfully detected. This completes the Western blotting procedure.
Just like Western blotting, a mass spectrometer can also identify protein, but mass spectrometer is a more precise tool as it can quantify protein based on their mass to charge ratio. Basically, mass spectrometer operates through 5 different stages, which is vaporization, ionization, acceleration, deflection, and detection. The first step occurs at sample vaporization chamber. The sample first enter the vaporization chamber, where a heating coil heats the sample and converts the sample from liquid into a gas. This is necessary because the samples need to be in a gas form before it can be ionized. The next step occurs at ionization chamber, where the vaporized sample moves into the ionization chamber. Here, an electron gun fires a stream of high-energy electron at the gas molecules. This knocks electrons off the molecules, converting them into positively charged ions. Any extra electrons that are involved in ionization are captured by electron collector, so they don't interfere with the rest of the process. These positively charged ions are now pushed through the ion acceleration chamber, and a series of negatively charged plates attract and accelerate these positive electrons, speeding them up and focusing them into a tight, fast-moving beam. The accelerated ion beam then enters the mass analyzer, which is curved and surrounded by a magnetic field. As the ion pass through, the magnetic field deflects their path. Lighter ions with smaller mass to charge ratio get deflect more sharply, while heavier ions deflect less. This is how the instrument separates ions based on their mass to charge ratio. Finally, the separated ions hit the detector, which generates an electrical signal proportional to the number of ions arriving. This signal passes through an amplifier to straighten it, and then it's sent to a computer, which processes the data and displays it as a mass spectrum, a graph of peaks representing the different molecules in the sample based on their mass.